my name is Lisa Offringa. I'm from the City University of New York and New York Botanical Gardens, and I'm here to present my dissertation research on the medicinal plants in northern Thailand used by traditional doctors to treat mild cognitive impairment in the elderly. My study was a traditional ethnopharmacological study with 4 different parts. So you start off with the ethnographic aspect of things, kind of an anthropological study, and then I went into the botanical aspect of my project, collecting the plants, testing them in the lab, and then I also did animal studies on one extract. The ethnopharmacological part of my research project also correlates with my dissertation project objectives. So these are the 4 objectives. I focused on the first 2 in a presentation last year at the SEB, and so for this presentation I'm going to focus on the 3rd objective. Just to clarify, mild cognitive impairment is one of the earlier stages of dementia. Memory loss is the most common symptom for this. About 15% of people with MCI get Alzheimer's disease, but because we weren't able to really clarify what kind of dementia the healers in Thailand were treating, we decided to go with MCI because it is the earliest stage of dementia, as opposed to trying to figure out like which type of dementia they were specifically focused on. My project looked for acetylcholinesterase inhibition plants. Acetylcholine is a neurotransmitter in the brain. It has to do with memory formation, and acetylcholinesterase is the enzyme that breaks down acetylcholine in the synapses of the neurons in the brain. And so what you want to do is you want to try to find plants that actually inhibit this enzyme. So by then inhibiting the enzyme, you have more acetylcholine in the synapses for the brain to access and for memories to be formed. They found that a decrease in acetylcholine in the brain happens with people as they age. And so in the elderly and in people with early stages of dementia, they found that increasing the amount of acetylcholine that's available to the brain will help remedy memory loss. So I'm going to quickly go through the first 2 objectives, which were the ethnographic portion of my research. I interviewed over 20 traditional Thai doctors. I interviewed them about Thai traditional medicine. I asked them about plant conservation, but mostly my interviews focused on what plants and plant formulas they were using to treat memory loss, and which plants they thought could slow or stop the progression of dementia. The second objective correlated with the botanical aspect of an ethnopharmacological study. I identified and collected all the plants in the memory formulas as well as some other plants, and then I selected 5 formulas. I worked primarily with 5 of the healers. I found that they had the most knowledge and information, were the most willing to work with me. So I took their 5 formulas and I tested them in the lab. Testing those 5 formulas in the lab wasn't really a part of screening for plants that will work as acetylcholinesterase inhibitors, but was more as a way to validate their traditional knowledge and be able to give that information back to them. So then what I did is I did a screen of the formulas. I took the plants that were that had already been tested, and I asked the healers to rank which plants they would test in the lab if they were doing this project, and so I came up with 12 plants to screen in vitro and in vitro bioassays. So I processed these herbal formulas. I did ethanol extractions, and then I did in vitro and in vivo bioassays. I started off by doing 4 different in vitro bioassays. I did 3 antioxidant assays. Antioxidants have been found to help in aging, to be neuroprotective, and to generally help people with longevity. I also did an acetylcholinesterase inhibition in vitro bioassay. So I'm going to start with the results for the antioxidant assays, and then go to the acetylcholinesterase assay. And so for the slides, I've set them up so that on this side is where the formulas are. Under here are all the individual plants. And you'll see that plant number 2 in all of these comes up as scoring the best in all of the bioassays. So that was one that I ended up choosing to test on animals. The DPPH is a very common antioxidant assay. In this case, it's an inhibition assay, so you want a lower number. So you can see that while some of the formulas also scored very low on this, plant number 2 was kind of a superstar in all of these. This is the FRAT bioassay. This is not an inhibition assay, and so in this case you want a higher number. So again, you can see where the formula scored really high. Some of the single plants scored really high. Number 2 still scored up in the highest range. Finally, I tested for antioxidants. I tested total phenolic compound. Total phenolic content. Phenolic compounds are a specific kind of antioxidant, and so this was a more specialized test to test those compounds specifically. It is, again, not an inhibition assay, so you do want a higher number. Plant number 2 and plant number 6 really just kind of blew the charts. So, but as you can see, plant number 2 is consistently coming up as the one that's scoring the highest. And then finally, the acetylcholinesterase inhibition assay. This is an inhibition assay, so you do want a lower score, and plant number 2, again, scored the lowest. So, here are some of the numbers for the tests. This is kind of an overwhelming slide, but the point of it is that, again, plant number 2 was chosen for the in vivo testing. So I did some rat behavioral models at the Faculty of Medicine in Konkan University. This is the experimental design. I had 6 groups with 8 young adult female Wistar rats. We treated them for 14 days. These are the different groups. There was a negative control where we didn't do anything to the rats, we just let them hang out. We had the vehicle where we gave them the solution that the extract was diluted into. We used Aricept, which is a pharmaceutical known acetylcholinesterase inhibition, and then we dosed 3 different doses according to their body weight and according to toxicological studies that had already been done on this plant. So I did 4 different behavioral models, but I'm going to focus primarily on the Morris water maze. This is the Morris water maze. It's basically a pool of water that's divided up into 4 quadrants. In each of the quadrants, there's a visual cue, so you can see like a yellow triangle or a red square, and then you put the rat into the water. You've also put talcum powder on the surface of the water, and into one of the quadrants you put a hidden platform. So you put the rat into the water, and you train them to swim around and find the platform, and you train them to do that, and then you start testing them on how long it takes them to find the platform, and so based on that, you have the first measurement, which is escape latency. Escape latency is how long it takes them to find the platform when they can't see it. So it is done in seconds. You time them in seconds, and in this one you can see that the top 2 doses, the 100 milligrams per kilogram body weight and the 200 milligrams per kilogram body weight, they showed a significant improvement in the time it took the rats to find the platform. Retention time is a measurement of how long the rats will swim in the quadrant where the platform is. So in this test, you remove the platform from the pool, and then the rats will go and swim around, and they'll swim around in the one quadrant, kind of saying like, wow, I thought the platform was here before, where did it go? So you test them on that, and it's how long out of, I think it's about a minute, maybe a minute and a half, how long they actually spend in that quadrant looking for the platform that once was there. So again, you can see the 100 milligram and the 200 milligram showed significant increase in the amount of time they spent in that quadrant. So those were the behavioral tests. Then after harvesting the rats, I took the 3 parts of the rat's brain and homogenized them with a buffer. I then tested them for acetylcholinesterase inhibition and oxidative markers. I'm only going to talk about the acetylcholinesterase inhibition test for the brain homogenate. So for this, in the hippocampus, which is one of the places that's responsible for memory, the middle dose showed significant activity. Again, sorry I didn't say before, this is an inhibition assay, so you do want lower numbers. For the striatum, the lowest dose showed significant activity. The striatum is a part of the brain that deals with focus and motor activities, so it's kind of how well they could focus on it and whether or not they could remember how to swim, or how well they remembered how to swim. And then finally the cortex, which is also involved in memory formation, the lowest and middle dosage showed activity. So in conclusion, for the in vitro and in vivo testing, plant extract number 2 was found to be an acetylcholinease inhibitor in low to medium doses in the in vitro tests and in the enzyme inhibition levels in the 3 parts of the brain tested. In the behavioral tests, it showed memory enhancement at a medium to high dose. And then it was also found, although I didn't really show these results, that plant extract number 2 exhibits some free radical scavenging activity, and also came up positive in many of the oxidative marker tests in the brain tissue of rats. So I wanted to talk a little bit about ethics, because this was an ethnopharmacological study, and ethics are a huge part of dealing with these kind of projects when you are in another country. So I obtained my research permits and my visa before beginning my research. I had to put together a number of institutional agreements between the institutions that I belong to and am affiliated with and those in Thailand. I worked with the government for about 9 months to try to get my permits to work here. And I got prior informed consent and IRB approval in Thai from all of the healers that I worked with. And finally, I'm working to protect their traditional knowledge. As you notice, all the plants were coded during testing, and all the plants are continuing to be coded until the results are published. At that point, I will release the names of the plant that I tested. This was part of the stipulations of my research permits with the government of Thailand. And I'm also including the traditional doctors in my scientific publications to make sure that they get recognition for the knowledge that they gave them for their lifetimes. And then finally, the next part of my research is to take my research results, put it into a formal document, and bring it back to the communities that I worked with. So I'm doing that in the next couple of months. I wanted to thank my collaborators in Thailand who have been kind and helpful more than I ever could have imagined. The Queen's Stericate Botanical Garden, the National Research Council of Thailand who gave me my research permits, Konkan University was where I did my animal behavioral tests and my in vivo tests on the rat brains, and then Chiang Mai University who offered me in vitro laboratory space. Finally, I'd like to acknowledge my advisory committee and all of the people that helped me along my way. My research assistant, my funders, and most especially the Thai healers who contributed to my work. I want to thank you all for listening, and I want to also just say that I think that medicinal plants really have given us so much and really have so much to offer us, and I think it's important not only to study the ethnopharmacological aspects of them, but also to make sure that they're protected and make sure that there are sustainable resources available for everybody. Thank you. Any questions?
Any questions?
Lisa.
Yes.
So the people, doctors you work with are being acknowledged in the report, right?
Yes.
And did that cause the Institutional Review Board to question you as why you would let their names become public?
I asked them if they wanted to be part of it.
Yeah.
I asked the healers. Specifically, I said, I'm publishing this, would you like to be a part of it?
So the Institutional Review Board had to acquiesce to their wishes, essentially.
I will assume so, to be honest. I haven't asked them, but I will.
It's an interesting predicament.
It is, and not one that I thought of.
After all, it's their intellectual property and they own it.
Right.
And they let you have access to it.
Right.
So no Institutional Review Board should be able to say no.
You would think not.
That's right.
Okay, thanks for bringing that up.
Can you repeat what the question was?
I asked if, since she was going to have the names go out of the doctors in the article, did that present any special problems with the Institutional Review Board? Because under the terms of that protocol, you should not be releasing names of informants. But she asked the doctors if they wanted to be a part of this. And I said, that trumps really the Institutional Review Board's situation. But some universities don't get it, and others do.
Thank you.
So it's a major question for many of the graduate students doing research to make sure the university actually does what it should do from the standpoint of the ownership of that intellectual property.
Lisa, you mentioned mixtures at the beginning of your talk. Will you be pursuing further studies looking at the role of these plants in a synergistic composition? Because you're looking at individual plants in this study, correct?
I would love to, if it would be possible. So, the answer to that is yes, if possible.
Yeah.
I would, because it's really interesting the way that they work together, and I found that one of the most fascinating parts of this work was just that there were so many formulas and then the crossover between the formulas and things like that. It was really interesting.
Great. Okay, so we're supposed to have a break now, but because we're running 15 minutes behind, I'd like to continue. That way we end on time. So we'll have our maybe 2 minutes for the next part of the session speakers to load their talks.